Hey guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will talk about D and L isomers. And if you are new in my channel, please subscribe my channel, and if you like my video, please do like, comment and share my video. So first of all, D and L isomers are mirror images of each other. So what do you mean mirror images? Like if you have a compound that is like A, B and C, that means the atom A has two bonds. So one bond is attached with B and another bond is attached with C. Now here you can see the C is now in this side and B is now in this side. So the compound remains same in both the cases, but the position of B and C is just opposite. In this case, we can say that these two are mirror images of each other. Okay, so let's check an example like D-glyceraldehyde and L-glyceraldehyde. So here we have D-glyceraldehyde and here we have L-glyceraldehyde. Now the thing is that you have to find out the chiral carbon first. Chiral carbon. So what is chiral carbon? So chiral carbon is such a carbon where the carbon has four different groups. Like the carbon, we know it has four bonds, right? Now here it has A, here it has B, here it has C, here it has D. That means this carbon is chiral because the carbon contains four different groups. Okay, so in D-glyceraldehyde, we can see that this carbon number 2 is chiral because it has OH in one side, H in other side, this CH2OH in another side and the aldehyde group CHO in the other side. So this carbon number 2 is chiral carbon. Here you can see the OH is attached in this right side and the H is attached in the left side. So this is D-glyceraldehyde. Now if you see L-glyceraldehyde, you can see that the OH that was in the right side in D-glyceraldehyde is now in the left side in L-glyceraldehyde. And H which was in left side in D-glyceraldehyde is now in the right side in L-glyceraldehyde. So these two are mirror images of each other and these two are isomers. Okay, let's see another example that is very famous example of glucose, D-glucose and N-glucose. So again, first you find out the chiral carbons. So we can see that carbon number 2, carbon number 3, carbon number 4, carbon number 5 are chiral carbons. The carbon number 1 and carbon number 6 are not chiral carbons because carbon number 2, 3, 4 and 5 all have four different groups attached with them. Okay, so in D-glucose we can see the OH is in the right side and H is in the left side in carbon number 2. In L-glucose you can see the OH is now in the left side and H is now in the right side. Now you see the carbon number 3, it has H in the right side and OH in the left side. But in L-glucose, now the H is in the left side and OH is in the right side. In D-glucose, in carbon number 4, OH is in the right side and H is in the left side. In L-glucose, the OH is in the left side and L is in the right side. In carbon number 5 of D-glucose, OH is in the right side, H is in the left side. And in case of L-glucose, the OH is in the left side and H is in the right side. That means the arrangement of this OH and H around the chiral carbon is different or opposite in D-glucose and L-glucose. Hence, these two are D and L isomers. So, it can be said that D-glucose and L-glucose differ in configuration at each chiral carbon. Now, this D-glucose can rotate the plane of light to the right side, that is in the clockwise side, while the L-glucose rotates the plane of polarized light to the right side, that is in the anti-clockwise side. And one more thing is very important to know, that D-glucose is found naturally in our body, but you will not get L-glucose in our body. L-glucose is only like you can make it artificially, you can make it in your laboratory. It does not occur naturally in your body. Do you know why? Because our enzyme machinery of cells can act on D-glucose only. Because we know that the hexokinase, that is very important enzyme, that is the enzyme of the glycolysis pathway. So this is the first enzyme of glycolysis pathway. This hexokinase and all other kinases, all other enzymes can break down only D-glucose. They can act on only D-glucose, but hexokinase cannot act on L-glucose. Hence, in our body, in all the cells, you will get only D-glucose, not the L-glucose. Hope you understood the concept. Thank you.